Hello students, welcome back to a new video of Sunanda's tutorial, and in this video we will discuss about apoptosis or programmed cell death. So it is an energy dependent biochemical mechanism of programmed cell death. So here one cell is destroyed or dead, but in a controlled manner, in a programmed way. It is genetically controlled process of deliberate suicide by an unwanted cell in a multicellular organism. So, this type of event occur during embryogenesis, metamorphosis, and tissue turnover. So, this type of process occur throughout the life cycle of an organism. It is the capacity of a cell to respond to a stimulus or a signal by initiating a pathway that leads to its death, okay, by a characteristic set of reactions or called as cascade reactions, okay. So this process of apoptosis is quite different from that of necrosis, because necrosis is energy independent cell death, okay, and that necrosis is the result of acute tissue injury, but apoptosis is energy dependent process and carried out in an orderly manner and genetically controlled manner. For example, apoptosis during the differentiation of human fingers in a developing embryo. Okay, during that process, the cells between the fingers to initiate have to initiate apoptosis so that fingers can separate from each other. So, these are our fingers. So during the process of apoptosis, the tissues which are present between these fingers have to destroy by the process of apoptosis so that all the fingers can separate from each other. The death of the cell by apoptosis is marked by a well-defined sequence of morphological change. So apoptotic cells become more compact, blebbing occur that at the membranes, chromatin becomes condensed, DNA is fragmented, and these cells shrink, condense, then fragment, releasing apoptotic bodies, which are generally phagocytosed by surrounding cells. Okay, so there are some chemical changes that occur on the surface of the apoptotic cells. Okay, so one of the important event that occur in the plasma membrane of apoptotic cells. So the negatively charged phospholipid phosphatidylserine, okay, is generally located in the inner leaflet of lipid bilayer of plasma membrane, but it flips to the outer leaflet of apoptotic cell, and where it serves as a marker of this cell. So other cells can easily detect the apoptotic cell and then kill them or eat them, okay. So then the mechanism of apoptosis. So there are three pathways by which apoptosis occur. One is extrinsic or death receptor pathway. Second one is intrinsic or mitochondrial pathway. Third one is porphyrin or granzyme pathway. Okay. It induces apoptosis by granzyme B. Okay, so it is three major pathways of apoptosis. So one of the most important enzyme which help in the process of apoptosis is caspase, and this caspase have proteolytic activity. Okay, caspase is otherwise known as cis or cysteine dependent asparated specific protease because they contain a key cysteine residue in catalytic site, and they selectively cleave proteins at the sites just C terminal or carboxyl terminal to asparated residue. So caspase is the main enzyme helps in the process of apoptosis. In these processes like extrinsic process pathway or intrinsic pathway and porphyrin or granzyme pathway. So here caspase generally plays important role. Okay, so at first caspase dependent extrinsic apoptotic pathway. So a number of extracellular signal molecules induce apoptosis. Extracellular signaling molecules means the signal or any signal that come from the exterior of the cell. These extracellular signal molecules bind with the cell's surface receptors, and those receptors are called as death receptors. One of the typical example is FAS receptor or FAS. These are member of TNF receptor family. TNF means tumor necrosis factor receptor family. Its ligand is FAS-L. Okay, its ligand means to which the receptor can bind. Okay, so the interaction of this ligand and FAS receptor on the target cell triggers apoptosis. When FAS bind to its ligand FAS-L, an adapter protein called FAS associated protein with death domain, which is called as FAD. FADD, FAD. Okay, FAD is recruited and binds to FAS, followed by recruitment of procaspase 8. Okay, so next FAS contains two domains, one death domain that bind to death domain on the FAS receptor, and one dead domain. Dead domain means death effector domain that binds to procaspase 8. Okay, the association of FAS associated protein with death domain with procaspase 8 results in the proteolytic cleavage of procaspase 8 to its active form caspase 8. So ultimately from procaspase 8, caspase 8 will be formed. Okay, and this activated form of procaspase 8 or caspase 8 molecule activate the downstream procaspases to induce apoptosis. So next processes will be carried out by this caspase 8, and it can conduct the apoptotic pathway, or apoptosis ultimately leads to the death of that cell. Okay. Okay, next is the caspase dependent intrinsic apoptotic pathway. So apoptosis is not only induced by the exogenous or external signals, but also it can be induced by intracellular death signal. This type of induced death cell has therefore been called as apoptosis by neglect as counterpart to the receptor mediated apoptosis by instruction. So in intrinsic apoptotic pathway, mitochondria plays important role. Okay, so from the injured mitochondria, a cytochrome C is released. Okay, then cytochrome C binds to the adapter protein called APAF1. Okay, what is the full form of APAF? That is apoptotic protease activating factor. Okay, apoptotic proteases activating factor. Okay, this interaction between APAF and cytochrome C leads to the hydrolysis of ATP to form ADP. Okay, this make a conformational change in APAF. Okay, then this APAF bind to the apoptosome. Okay, then this apoptosome, formation of this apoptosome then recruit another enzyme called procaspase 9, and procaspase 9 finally forms caspase 9. Okay, this caspase 9 in turn form procaspase 3, and procaspase 3 then activate to form caspase 3. So what we have learned that at first procaspase 9 form caspase 9, caspase 9 form procaspase 3, procaspase 3 form caspase 3, which causes the apoptosis of the target cell. Okay, so this intrinsic apoptotic pathway actually regulated by BCL2 family of proteins. This family of proteins comprises both anti-apoptotic member which inhibit the apoptosis and pro-apoptotic members which activate the apoptotic pathway. So anti-apoptotic members are like BCL2, BCLX. These are anti-apoptotic factors. Okay, but there are some pro-apoptotic member which promote apoptosis, and those are BAX. Next, BAD, that is bad, that is another factor is BIC. Okay, these are promote apoptosis. Okay, and BCL2, BCL stands for B cell lymphoma or leukemia 2 gene. Okay, so this BCL2 family of protein control or regulate the release of cytochrome C from the mitochondria. The anti-apoptotic BCL2 protein actually they inhibit the apoptosis. Okay, the pro-apoptotic BCL2 protein consists of two subfamilies. What is pro-apoptotic? Pro-apoptotic factor means they promote the apoptosis, but there are some anti-apoptotic member or factor, they inhibit apoptosis. Pro-apoptotic members are like BH123. This is one subfamily of BCL2, pro-apoptotic BCL2. Another one is BH3. Okay, these are pro-apoptotic BCL2 protein subfamilies. Okay, back these are, but there are some BS3 proteins like BID, BIM, BIC, BAD, PUMA, NOXA. Okay, so those are pro-apoptotic BCL2 protein subfamilies. Okay, so pro-apoptotic means they promote apoptosis to occur, but some anti-apoptotic factors are there like BCL2, BCLX. Okay, so in this way they control or regulate the intrinsic apoptotic pathway. Okay, another pathway of apoptosis is granzyme mediated pathway. So here one protein that is porphyrin make a pore or hole on the cell membrane of target cell, and it activate the entry of granzyme B inside the cell. Granzyme B will activate caspase 10 inside the cell. Caspase 10 in return activate caspase 9. Then caspase 9 will activate caspase 3, and it will lead to the DNA fragmentation of the target cell, and ultimately the cell death occur. So in this way these three pathway conduct the apoptosis of the target cell or unwanted cell of the body. Okay, So it is all about apoptosis, different pathways of apoptosis. I hope you really enjoyed this session and will get benefit from this. And if you liked it, please hit the like button, please share with your other friends, and please subscribe to my channel Sunanda's Tutorial to get more videos like this. Thank you.